So hi, hello and welcome again, Microbehunter here. A few days ago I found this water sample quite accidentally in a plastic bottle, and of course I wanted to now share with you my finds, or at least what I'm going to hope to find, because I don't know exactly yet what is in here. Well, I do know a little bit. I had a quick check just a minute ago, but maybe there are some more interesting organisms that I did not see yet. I'm first going to play the clip on how I actually found this water sample, because the place was rather unique. Yeah, and there is something very interesting that I found here. Look, I cannot believe it. Look, 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 look. Ah, what is this here? Ah, ooh, haha. Yeah, of course, standing water like this is, of course, a problem, because this is a breeding ground for mosquitoes, and therefore, yeah, it should be removed in any case. Yeah, okay, so here I am again. In order to put a small sample from of the algae here on the microscope slide, I think I'm going to cut off the tip of the pipette here, because I would like to have a slightly larger opening here, and then I'm not just going to take some water, but actually I would like to take some of the green stuff here as well. Plenty of algae are in here, of course, and usually where there is some solid material, oops, I did not catch anything here. Usually where there's some solid material, there are also some other microorganisms there that sometimes attach to the surface. Yeah, a small amount. Usually it's a good idea to kind of resuspend it a little bit to break apart the clusters. Okay, yeah, and then, I think I'm just going to put a cover glass on top, and then we're going to see. Okay, so here we go, and it goes under the microscope. Yeah, usually it's always the same suspects that I'm finding here. Of course, a Vorticella, quite a common ciliate. It's growing on a stalk, and maybe if we're lucky, we're able to actually see it contract. I already made several videos about this one here. Of course, the many different types of species, they all kind of look similar in the sense that they are growing on a long stalk, and they have a ring of cilia, these are tiny little moving hair on the top. It's a one single cell, right? And actually I want it to contract, and if it doesn't do that, I'm just going to tap the microscope slide a little bit, and maybe this little shock will actually cause it to contract. So I'm going to use my tweezers here, and I'm going to, yep, that was it. And this is going to start again up, again. Yeah, usually if it touches something here, up, so yeah, it started to touch. Maybe you've seen it, there was this little thing there that it touched. Yeah, and here we go again. Maybe I'm able to do it again. I'm going to tap. See? Yeah. I think you get the idea. Yeah. So as a matter of fact, the contraction of the Vorticella is actually one of the fastest movements known in biology. I'd be careful to say the fastest one. One has to be careful with superlatives always. But indeed, it is a very fast and very rapid movement. It's a form of protection. And what it does, and let me switch on the arrow again here. When I turn on the arrow, then I'm able to actually point now, of course, to this tiny, ring of cilia. These are beating here over here, and it's called Vorticella, of course, because it produces this vortex of water in here. But let's look for some other microbes in this sample. Yeah, let's scan the slide a little bit. Of course, many algae, possibly chlorella, because the round algae often are chlorella. It's a very common one. Yeah, there is something moving over here. Let's zoom in here a little bit. Yeah, this guy here is pretty deformed. Yeah, it's clearly also a ciliate, which with its little hair around it, it's a little too difficult to catch. And the water sample under the microscope slide is actually fairly thick because of the algae, so it's going in and out of focus quite rapidly. This round circle that you see here, that is an air bubble, or rather an oxygen bubble, because the algae are doing photosynthesis, and if I now were to wait for several minutes, then we'd be seeing how the oxygen bubble actually starts to grow in size as more and more of the oxygen accumulates, which is formed by the algae by photosynthesis. Yeah, what's this dark part here? Nothing. Yeah. So I'm going to be looking again around a little bit for something else that's interesting, and maybe we're going to revisit this oxygen bubble in a couple of minutes. Yeah, I found two more Vorticellae growing right next to each other. They have just started to contract. Maybe they're going to extend again. Yeah, they bumped into each other, and now Yeah, they don't want to go apart anymore. They don't have a lot of space really. Yeah, but let's see. Let's be a little bit patient. Maybe they're going to extend. Yeah, it's difficult. Yeah, not enough space, and as soon as they touch an object, they quickly start to contract. And you can see, of course, here quite nicely as well that this one over here is again looks a little bit different than the one that we just saw. Yeah. So, but maybe there are a couple of other things that I would like to show you here. Let's switch on the arrow again. Yeah, and you can actually see the cell organelles in here, and yeah, basically it creates this vortex of particles of this water stream, and then it sucks in, so to say, by it engulfs. Engulfs is the wrong word, but it essentially takes in those food particles, bacteria, and other debris to form a so-called a food vacuole, and then this food vacuole inside the cell is being digested further. Yeah, but this one here is a little bit of a more of a difficult one because yeah, it's a little bit very crowded here. So let's move on and let's have a look at for another one. Yeah, look at all these oxygen bubbles that have formed in the meantime. Yeah, the algae are producing a lot of oxygen. And of course, many microorganisms in here are going to be quite happy about that, about this oxygen supply, because those moving organisms, of course, they need a lot of oxygen because the oxygen is necessary for cell respiration to release energy so that they can actually do the movement. Yeah, and here is yet another one, again, with a slightly different shape. Let's tap the slide again. Or let's try the cover glass up. This was a little too much. Okay, this was a little too much. Okay, now I think it, yeah, up. Now it got stuck here beneath this long fiber. So this here actually is a nice find. This one is a so-called a Heliozoan, also sometimes known as sun animalcules. It's not an animal, of course, but the name kind of sticks ever since Anthony van Leeuwenhoek back a few hundred years ago called these little critters animalcules, all these microorganisms. And those spikes that you see are actually called axopodia, axopodia, and it uses them to catch prey. So I've once actually seen that a cell got stuck here and it was slowly drawn towards the body, right? And then it was somehow digested, devoured. Yeah, they don't move on their own, really. They just float around. But look, there is some kind of a movement coming here from the bottom. I'm going to check where this stream of liquid is coming from. Yeah. Look at this. What is this here? Okay. Yeah, this here is this gigantic one here is a Rotifer, right? Look, yeah, it's green on the inside here, probably because it's eaten, yeah, a lot of green algae. Rotifers are, yeah, it's now a question of debate of whether you want to call them microorganisms or not, because these are multicellular animals. So these are true microanimals in comparison to, let's say, the Heliozoan, which is a single-celled microorganism here, right? That's even though it's called animalcule, yeah, it's actually not an animal. Yeah, this one over here actually is, right? Because it has it's composed of many different cells. It has a foot over here with which it likes to connect to a surface. But right now we don't see it quite well because it kind of is stuck in here a little bit. So what I'm going to be doing is I'm going to be adding a little bit more water to lift the cover glass a little bit so that we're able to see it a little bit better of how it looks like. So I'll be simply adding a little bit of water now. So you can now actually see the stream of liquid, of water, yeah, now going the other direction here. Yeah, and maybe if we're lucky, it will give it a little bit more space. Yeah, look at all that liquid streaming by. It also relieves a little bit of the pressure now from of the Rotifer. Let's see if it's able to crawl out. Yeah, despite the try, it seems to feel quite comfortable in there and doesn't want to come out. Okay. Yeah, unfortunately, I just dipped my objective in a little bit of the water, so I had to clean it again. And in the meantime, I found this one over here. I don't know if it's the same one that we've just seen before. In any case, that's a Rotifer. It looks a little bit, quite a bit longer actually. So it might be a different one. I don't know. Yeah, unfortunately, I had to remove the slide to get my objective clean, so I don't know exactly where I was before, and everything got shifted a little bit, but at least yeah, we're able to see a Rotifer a little bit better now. Well, I switched now to phase contrast and I found another Heliozoan, and now we're able to see those axopodia much better, those tiny little spikes, because they appear a little bit darker than the background. So yes, I think that's also a very unique little organism here. I think the beauty of nature, I think that's all I can say here. It's fascinating for me to see the vast diversity of biodiversity of different organisms that you're able to find in just such a simple bottle of rainwater, which has been in nature for maybe a couple of weeks or months. It has given it enough time to yeah, for all of these organisms to reproduce. And if you ever have a water sample that you would like to look at, then I recommend let it stand for a couple of days at home, because sometimes this will give it sufficient time for the microorganisms to reproduce, and then you have a natural progression of different microorganisms over time. I am just going to call it quits for now. The air bubble, the oxygen bubble up here, you can actually see how it's actually growing. Yeah, so you can see that the sample is biologically very active. Yeah, and it's growing. Yeah, but for me, that's all I would like to share with you today. Happy microbe hunting as always, and see you in the next video. Bye bye.